Okay, so the male reproductive tract. So hopefully we're all on the same page. Hopefully through reading a lot of this makes sense. But there's a lot of details, a lot of specialized cells doing certain tasks. And so we're going to go ahead and run through that. So just starting off, right, making sure that we're all clear that the male gonad is the testis. The pair of them then are testes. The main male hormones are the androgens, and the primary androgen then is testosterone. So androgen is a whole class of hormones, but testosterone is going to be the most prevalent. And then, of course, the gamete that they make are the spermatozoa, or the sperm. And so we're going to look at how all of these structures and hormones and cells work together. So the testes, and we were doing some dissections in lab, so a little awkward that lab kind of came before lecture, but then hopefully a lot of this anatomical terminology is very straightforward for us then. So remember that the testes do need to stay outside of the body, right? The ideal temperature for sperm formation is slightly cooler than body temperature. So in Celsius, if body temperature is 37, I think they say 35 degrees for sperm production to occur properly. So when you look at the testes, right, they're housed in this sac called the scrotum, and that does have some muscle inside of the scrotum, and they point out it's the dartus muscle and the cremaster muscle, and those are going to be important for basically regulating the elevation of the testes, so they can contract and pull the testes closer to the body to keep them warmer, or relax and let the testes move further away from the body to cool off, right? Again, trying to maintain kind of that ideal body temperature or ideal temperature for sperm formation, which is not the same as the ideal body temperature. So they adjust to the environment. Now, one of the most fascinating things I find on the male reproductive tract is that during development, and so this is kind of a hard picture, right? This is saying at approximately 8 weeks post-conception, right? This is really early on during development, we see that the testes are actually up in the abdominal cavity. And so what's really going on here is that basically in early embryonic development, the testes and the ovary are the same. And it's going to require basically some of the proteins that are produced due to the chromosomes the genes on the Y chromosome are going to start moving that baby towards becoming male. And so what's going to have to happen is this organ needs to differentiate into the testes. And so we're going to see it start to descend, start to move down through the body. And so here they're trying to show that it's a little awkward picture, but I do. Right? Remember we saw this in lab, the ductus deferens or the vas deferens, the way it was really close to the bladder and kind of wraps around the urethra. And we're all like, why is it doing this? It starts to kind of make sense if you know the embryology of this, right? So the testes starting up really in the abdominal pelvic cavity, close to the kidneys, and then descending. So between 7 and 9 months before birth, 7 to 9 months post-conception, so just before birth, these testes should be fully descending, actually leaving the abdominal pelvic cavity through the inguinal canal and moving out into the scrotum. So they need to descend, right? And there's a disease would be what we'd call it, called cryptorchidism, which is when one or both of the testes fails to descend. And this can increase rates of infertility and also increase rates of testicular cancer. And so that's something that's typically going to be corrected surgically. Again, this is a picture from another book. I thought that was kind of interesting. Again, they're showing this baby is tiny at this point, right? And actually showing how the testes move down. And then this front view, I think, was really helpful as well. So we see the gonads way up by the kidney, and now we're actually calling them a testis, right? And they're descending, moving through the inguinal canal and out into scrotum. The other thing that this leads to, though, right, and a few of us talked about this in lab, right, is that the testes have to be able to get out of the body. So you have this inguinal canal, this opening, this tunnel that the testes and then the spermatic cord kind of follows to get out of the abdominal cavity. And so in males, then, this whole, how do I want to say this? The surrounding tissue maybe is a little bit weaker. There is like an actual opening in females, since the testes we don't have testes, don't move through there, right? It doesn't get enlarged the same way. And so males are more at risk of an inguinal hernia. And really what we're looking at there is intestinal contents, particularly the small intestine, pushing through that inguinal canal and protruding into the scrotum. And the big problem here is that you can actually cut off circulation to that portion of intestines. It can die, it can become necrotic, and so on. And so that would be an inguinal hernia, right? In the digestive tract, we talked about a hiatal hernia. This would be an inguinal hernia. And so that can get corrected surgically. They'll go in and put like a little mesh over the inguinal canal to make sure, or to try to limit that from happening. Excuse me, from happening again. Ooh. Okay, so then we are going to jump in here into spermatogenesis, and we looked at this histology in lab, right? But keeping in mind here, right, the testes are filled with these tubes, right, the seminiferous tubules, and this is where spermatogenesis, the production of sperm, is going to take place, right? And each testis has about a half mile worth of seminiferous tubules. So we're seeing a big importance here of producing sperm, right? And we're giving lots of surface area for those developing sperm to be nourished and develop. So just as we're zooming in here again, this is kind of what we were looking at in lab. And then a cartoon version of the same thing, right? So noticing outside of the seminiferous tubule, and this area labeled as the lumen is going to be then how sperm actually leave the seminiferous tubules, move into the epididymis, and eventually the ductus deferens. Cartoon. Okay, so this is the slide that I want to work off of, and then I'll show you the book as well. The book was good. I just like this one a little bit better. Okay, spermatogenesis. So we're in the seminiferous tubule, and what we're going to see here is that, let's see, where's my spermatogonia? There we go. So these cells at the very outside of the seminiferous tubule, right? So the most superficial, the furthest from the lumen, right? These are called spermatogonium. Spermatogonium is the stem cell, right? So this is a cell that can keep dividing, and therefore what we'll see happen, right, is I like this part right here. So the spermatogonium, when it does mitosis, right, it's a stem cell, so it divides into two. One of those two is going to move forward in this process of sperm production. The other is going to stay behind to remain a spermatogonium that can then divide, right? And one of those might go on, and the other daughter cell remains behind in order to undergo mitosis. So it is important to realize that it's both mitosis and meiosis involved in this process. That is why males can continue to produce sperm pretty much throughout their lifetime, because the spermatogonium does mitosis, and then one of those daughter cells goes on to become sperm. The other spermatogonium does mitosis, and one of those goes on to become sperm, okay? So this first step right here, where I was drawing these little arrows, is mitosis. So one of those daughter cells produced by mitosis, then that's destined to become sperm, is called a primary spermatocyte. The primary spermatocyte is going to undergo meiosis 1 to become a secondary spermatocyte. they're not showing that very well here, right? I want to see two daughter cells right here. So a primary spermatocyte does meiosis 1, and the offspring, the daughter cell of that is a secondary spermatocyte. Secondary spermatocytes are then going to undergo meiosis 2, right? So secondary spermatocyte in meiosis 2, and then that's where we're going to end up with our 4 haploid spermatids. So spermatid is still like an immature, it is not yet sperm, right? It's small, it's not specialized. I think this is really weird that they drew 6 of them, right? Because there'd be 4 daughter cells if we were tracking a single primary spermatocyte going through this division process. Okay, and then just a reminder, right? So if this is going through meiosis 1, we split the homologous pairs, right? So the secondary spermatocyte is already haploid, and then to become a spermatid, when we divide again, we're separating those sister chromatids. So from there, right, we want to become actual sperm, right? And so these spermatids need to go through another process. This is basically the final process of spermatogenesis, which is spermiogenesis. Okay, notice that. It just caught me too, right? Spermatogenesis is how we're taking spermatogonium and going through meiotic processes to build sperm, right? Basically to get to spermatids. Then, right, spermatogenesis. Spermiogenesis is the final step, and it's the maturation of those spermatids into spermatozoa or sperm, right? The mature cells that are differentiated, right? Notice now these look totally different. They have really specialized and they're filling that final role. And we'll look at basically what happens here is there's lots of other cells. Maybe not lots. There are other cells that are involved in this process. So one of the things that we'll see, and we'll go into this in a little more detail, is that all of these cells at the end of meiosis are actually still attached to each other. They have little like cytoplasmic bridges attached to each other. Cytokinesis, the final splitting of a cell, doesn't really quite finish. And what we're going to see is we have these nurse cells, or I think your book also called them Sertoli cells or sustentacular cells are also names. So for making sperm, Sertoli, sustentacular, they all have S's, and then also sometimes they're called interstitial cells, nurse cells. So these nurse cells or Sertoli cells are going to be helping things out, and we'll spend a bit of time talking about them. They're really important in how this process works, and particularly spermiogenesis. They're going to go through and actually pull off like the extra cytoplasm, like make these guys nice and lean and ready to go. Right? So then this is that same, essentially the same image from your book. Notice here is the outside of the seminiferous tubule. This is the lumen, so it's flipped over here. But it's the same thing that they're showing. Again, the stem cell, the spermatogonium, undergoes mitosis to split in 2. One stays behind as a spermatogonium. The other daughter cell becomes the primary spermatocyte that undergoes meiosis 1 to become secondary spermatocytes, which undergoes meiosis 2 to become that spermatid. And then we need spermiogenesis to get us to the point of actually becoming sperm cells. Okay? And then again, here they're trying to show those sustentacular cells, also called Sertoli cells, also called nurse cells, that are going to help in this process of maturation. Okay. So when we look then at the actual spermatozoan, right, or sperm, it's a really unique cell in the human body, right? So the head of our sperm here contains the nucleus. This is where the genetic material, the chromosomes, are located. At the very tip here, we have what's called the acrosome, and this is like a little packet of enzymes right at the tip of the head that are going to be necessary for actually penetrating the ovum, right? So they're necessary for fertilization. Here in the kind of middle part, the neck of the mitochondria, we will see a lot of those little blue spheres. They're trying to show all these mitochondria, right? This guy needs to be able to swim. He needs a lot of ATP. And so we find this kind of central region of the sperm highly, very rich in mitochondria in order to make that happen. And then, of course, the tail. This is a flagellum, and this is the only flagellum that you will find in humans, right? Very similar if you look anatomically, right, which is very small, but at the anatomical structures inside of this flagellum, very similar to a cilia, right? If you think about cilia in the respiratory tract, they're moving in this wave-like motion, right? And we're going to see very similar proteins involved here in setting up the flagellum, but the only flagellum in the human body. Okay. Oh, that's the last thing I wanted to say here. So realize that just because it looks like a sperm, right, doesn't mean that it's really ready to go. So before the sperm are able to use that flagellum, before they become motile, they have to go through another process called capacitation. And this is when they actually become motile. And this occurs when they come in contact with the seminal fluids. That actually helps activate them, make them able to swim. And then they actually aren't able to cause fertilization until they become exposed to the conditions of the female reproductive tract. So this is all very, very carefully choreographed. Okay, oh, and so what you'll notice, that's why I pulled this, right, is what you'll notice is if you look at like the epididymis, the ductus deferens, you're going to actually find ciliated epitheliums lining these tubes, and it's the cilia, right, that actually propel the spermatozoa. So these spermatozoa that are hanging out in the lumen, right, in the center of the epididymis, they're not swimming yet, right? And so it's really the cilia on the cells in the male reproductive tract that help move them along. Okay. I wanted to tell you more about nurse cells. Okay, let's go back and talk about nurse cells then. So nurse cells have a lot of really important roles. So ready for this? So nurse cells. I've got 6 different things, I think, here. So, and I don't think your book went into great detail here. So a couple things. There is a blood-testis barrier, right? So notice this kind of dotted line here. Just like the blood-brain barrier, let me get rid of some of this writing if I can. Okay, so now we're looking at this blood-testis barrier. So just like there is a blood-brain barrier, we're really committed to not letting things affect the testes, right, and the development of these spermatozoa. So the nurse cells actually help create or maintain this blood-testis barriers. So there are tight junctions in the cells in this region that try to prevent things from getting through. And then the nurse cells are also helping out. So this really, we're attempting to isolate the seminiferous tubules from the general circulation. And one of the things to consider there is we're really trying to keep it away from our own immune system, which is kind of interesting. The cells here haven't gone through the same process maybe of training the immune system the way most cells in the body have, where they're kind of run through the thymus and screened. And so now we need to keep them separate from the immune system. And in fact, if you have like a bad testicular injury, that could actually kind of rupture this blood-testis barrier, and that can lead to some autoimmune kind of situations. Okay. They're also, the nerve cells are super important in maintaining the fluid composition inside the seminiferous tubules. So in order for this whole sperm production maturation to work well, you need the fluid composition to be different, right, than it is in this plasma outside, in the blood supply. And so the nerve cells make sure this fluid is perfect for sperm development. So it's higher in androgens, the testosterone, higher in estrogens, higher in potassium, higher in amino acids, right? Just the right conditions, okay? And so that's one of the roles of these nurse cells / sustentacular cells / Sertoli cells, whichever you want to call it. They should just decide. Okay. They, of course, then are supporting both the mitosis of the spermatogonium and the meiosis of the spermatocytes. And really, they're told to encourage this process by things like luteinizing hormone, by follicle stimulating hormone and testosterone, right? So they're just doing what they're told, but they are important in this process. They support spermiogenesis, which I mentioned earlier, right? This movement from this like non-distinct, haploid daughter cell blob to an actual mature sperm. And so they use nutrient and chemical stimuli as they're developing that on these spermatids, right, on these immature sperm. They do things like, I mentioned, phagocytizing the cytoplasm that's being shed there, right? And so they're kind of grooming them towards adulthood. Nerve cells also produce the hormone inhibin. I don't remember if we talked about inhibin with the endocrine system, but this is going to have a negative feedback. It goes back to the pituitary gland and inhibits follicle stimulating hormone release. And so, right, as you have a lot of sperm cells moving towards maturity, we're being told, okay, we have enough, right? So like ease off the follicle stimulating hormone, which would tell these nerve cells to encourage more division, right? And so it's just a negative feedback there. Nerve cells also produce a protein called androgen binding protein. This is one of the ways they're actually able to make sure that the interstitial fluids have higher levels of androgens. So they kind of help concentrate it in the seminiferous tubules. And then finally, number 6, in the developing fetus, nerve cells produce, it's called Mullerian inhibiting factor, but it basically, how do I want to say this? It gets rid of what would be fallopian tubes, what are actually called Mullerian ducts, because when you're developing, you actually have both, right? We're not sure yet whether we're going to be male or female, and so you have the Mullerian ducts, which would end up becoming fallopian tubes, and you have the Wolffian ducts, which would become the ductus deferens. And so the nerve cells produce this Mullerian inhibiting factor and basically tell the Mullerian ducts, the female ducts, to regress, to disappear, right? And so that's part of this developmental process to make this baby a boy. So a lot actually going on there in that development. Sorry, I'm flipping. Now I want this picture. Sorry, kind of out of order here. Because one other thing we don't really talk about a whole lot is the epididymis, right? So we found that in lab. They talk about it being this like comma-shaped structure that's kind of surrounding each of the testes. So what happens here is that once the spermatozoa are mostly developed, right, they're going to detach from the nerve cells and they're hanging out in the lumen of our seminiferous tubules here. They're still functionally immature, right? We said they can't actually swim. They get carried over to the epididymis and the ducts that we find in there. And again, they're carried just by fluid movement because they can't swim. And a few things actually happen in the epididymis, and I think of them as kind of hanging out there. They're finishing up their maturation process. In fact, your book points out that if you take sperm from the head of the epididymis, they're unable to fertilize an egg, and it's not till you get them down here in the tail that they've kind of moved through this process. So in where is the picture I am looking for? There we go, right? So then in the epididymis, this columnar, pseudostratified columnar epithelium is really monitoring the fluid composition, keeping that good still. It recycles. This is where any damaged spermatozoa would be recycled. And then here in the epididymis, they're being protected and allowing to mature further. So it's bad. I'm kind of like, it's like the hangout place, right, where the sperm are waiting. But they are going through some developmental processes here. From the epididymis, then, of course, they're moving into the ductus deferens. And this is going to be, again, they're being transported. They're not swimming on their own at this point. So it's actually going to be peristalsis, smooth muscle contraction, that's going to move the sperm and really until they reach the ejaculatory duct. Okay, so that brings us to all these accessory glands, right? So the sperm started out in the seminiferous tubules, went through their development, most of their maturation. They then hang out in the epididymis and then move into the ductus deferens. So when we decide it's time to go ahead and donate these sperm to a fertilization project, right? Those sperm are going to move up through the ductus deferens to become part of the semen. But really, the testes only contribute something like 5% of the semen, right, is actually the sperm. What we're going to see is that we have a bunch of accessory glands that jump in here as well. So again, the ductus deferens goes all the way until we get to what's called the ejaculatory duct, but this is where we start to see products of the seminal gland or the seminal vesicle. This gland produces about 60% of the semen. And what you're going to find in this glandular product is fructose. So this is going to be fuel for the sperm, right? They have all those mitochondria. They want to make ATP. Fructose is going to be the fuel for them. The products of the seminal gland are slightly alkaline, trying to help neutralize acid, both in the urethra, because remember the urethra also carries urine. And so you could use this slightly alkaline product to kind of neutralize that. And also the female reproductive tract is slightly acidic, particularly the vagina. We have lactobacillus that live there and are constantly producing acids to keep other microbes out. And so this is going to help protect the sperm as they travel. We'll also find in the seminal gland prostaglandins. These stimulate smooth muscle contraction. This is also where we release fibrinogen. So what's going to happen is you're going to have like a temporary clotting of the semen when it enters the vagina. So fibrinogen is the protein that we saw forming blood clots. And so it's going to have the same role here. The introduction to these fluids is also what finishes that maturation of the sperm. It goes through this process called capacitation, where now the sperm are able to swim, right? So the seminal gland or seminal vesicle is adding its contributions here into the ejaculatory duct, as is the vas deferens. The prostate gland, right, is where we're going next. So we're going to go into that prostatic urethra. The prostate gland contributes somewhere around 25% of the semen. It contains things like an antibiotic named seminal plasmin. So this is going to help protect the sperm, right? You produce antibiotics and antibiotics and you send it with the sperm. You're trying to keep them safe. The bulbourethral glands down here in the urogenital diaphragm, really small contribution, again, somewhere around 5%. And really they're contributing a thick kind of alkaline mucus, again, really trying to neutralize those acids and protect the sperm. So again, contributions of the testes, the seminal vesicle, the prostate gland, and the bulbourethral glands is then what is going to constitute semen. Okay? On average, somewhere between 2 and 5 milliliters of semen per ejaculate. That is going to contain, get this, 20 million to 100 million sperm, right? which of course you don't need all of them in order for fertilization to take place, but you do need more than one. And we can talk about that at the end. Once we've covered both systems, we'll talk a little bit about fertilization. So again, only 5% of the semen is sperm, but that's 20 million to 100 million sperm in there. The seminal fluid then, products of 3 different glands, again, containing enzymes, antibiotics, fibrinogen to make the semen plug. So they're actually fuel for the sperm. There's a lot going on there. Okay, and then just a reminder that we did talk about this a little bit in the endocrine system, but I thought this was a really nice kind of map. Remember, this is all started out in the hypothalamus with gonadotropin releasing hormone. That's going to tell the anterior pituitary to release luteinizing hormone and follicle stimulating hormone. Okay, luteinizing hormone talks to the interstitial cells, also called Leydig cells. These are in the testes producing testosterone. Okay, and notice the production of testosterone provides a negative feedback both on the pituitary and the hypothalamus. Follicle stimulating hormone is the one that talked to the nurse cells, right, the Sertoli cells, sustentacular cells, and caused spermatogenesis, right, sperm development. They also produce, remember we mentioned the nurse cells producing inhibin, and that is going to have a negative feedback on the pituitary gland as well. So when there's lots of sperm production taking place, we go, okay, hold on a second. But then as it declines, right, you lose some of these signals, and so we get more FSH and OH. We up the production of sperm cells. So just a negative, kind of a standard negative feedback loop. Okay, that is all I have to say about the male reproductive tract. We will be back shortly with the female reproductive system.